Drug developers, investors, researchers, and corporate executives wrestle weekly to understand what is happening in commercial development of NASH medications. Join hepatology researcher and key opinion leader Stephen Harrison, patient advocate Donna Cryer, liver wellness advocate Louise Campbell, and forecasting and pricing guru Roger Green as they discuss the issues affecting the evolving NASH market from their own unique perspectives on the Surfing the NASH Tsunami podcast.
For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up. Episode 31 of Surfing the NASH Tsunami starts now. This week is the calm before the storm. Last week was episode 30. Next week starts our AASLD coverage, which with 1 week off will run up through U.S. Thanksgiving last week in November. As soon as that ends, we're going to start talking about NASH-TAG and end-of-the-year wrap-up and visions for 2021. And yeah, somewhere in the middle of all that, for those of us who live in the States, there's this thing called the presidential election that might give us a little bit of excitement in one way or another. This is the calm week. And that's going to allow us to do two things I've always wanted to do. The first one is I always wondered if you could sell products on a podcast. And as Donna reminds me, this week is October is for Livers Month from GLI. And she would like to talk about some rather amazing clothing, wear, and accessories that they put out as part of the month. Donna, tell everybody what they can win or buy.
Thank you so much for the opportunity. First of all, I really want to applaud all of the advocacy organizations around the world in Europe, in the UK, in the United States, who are coming together to raise awareness of liver disease and liver cancer during the month of October. I also give a nod to my sisters in the breast cancer space for the way that they have paved and for the success and improved outcomes that they have for their patients. So we have a lot to learn here in the liver community. But yes, the Global Liver Institute's campaign is hashtag October is for livers, use the number 4. And we had the opportunity to have a set of t-shirts designed for us by an artist, author, Christina Hagman, who is the daughter of the actor Larry Hagman, who had a liver transplant and has passed. And based on her family experience with transplantation and cancer, she designed a fantastic image for us, emerging from the story and the memory that she holds being pregnant with her child while her father was nurturing his new liver. So the image of a liver being nurtured and is really significant to her and significant to us. And as we know, during this pandemic, liver cancer can't wait. Neither can liver patients. And so if people would like to support the campaign, they can find information on the globalliver.org website. There's also a fun auction going on if you're a Hamilton fan and want to take dance classes directly from the cast. So yes, so now my team is happy with me. I am wearing the t-shirt, although it's a podcast, so you can't verify that, but they are comfortable and fantastic, and I will be wearing it and other things, all GLI related, for the rest of the month. Hope you will too.
Donna, if you would like to take a picture of yourself wearing the t-shirt, and you said that you and your husband did an impromptu fashion shoot on the deck yesterday, we'll be happy to post that on the webpage along with the episode and a link to the other clothing and paraphernalia so people can see what it looks like on you and on Dennis and then buy whatever they want to. If you don't send the photograph, then we'll just post the website.
Thank you. That will help get my aspirations to be a 50-year-old Instagram model.
Perfect. And in fact, that could be one more thing that transplant people do that no one even recognizes. They become fashion models at age 50. I love it. Okay, so that's one. The second thing we don't do often around here is we don't talk about on-the-ground patient care, right? This is a podcast. It's pretty much focused around a journalistic enterprise, so we focus on what's novel, new, noteworthy, all those N-words. What we don't do normally is talk about the things that happen every week, everywhere, which is doctors taking care of patients. It felt like a really good time to stop and ask, what does that look like? And how does that relate to the kinds of things that we talk about on this podcast? I thought it might be a particularly good idea to do that in the UK rather than the US, because the UK is witnessing a couple of initiatives that are bigger than or different than anything we're seeing in the US right now, rather than having executive leadership that questions whether medical advice around the treatment of pandemic viruses is sound. The UK is now embarked on a campaign around obesity, which might have had something to do with experiences some of its leaders had earlier in the year, but to recognize exactly how big an issue obesity is and for the government to get behind educating and dealing and managing, number one. And then the British Liver Trust came out with a campaign called Sound the Alarm about a liver, which is the kind of thing you would expect GLI to do maybe, but on a slightly more dramatic scale because of what British Liver Trust means in the UK. So for those reasons, I I turn to Dr. Ian Rowe, who is a good friend of the podcast, someone who comments on LinkedIn and Twitter, and who I chat with from time to time to make sure we're not getting way off base in what we're doing. Spends half his time in clinics with patients. So Ian, welcome to SurfingNASH.
Thank you, and it's great to be here. I think I've listened to every episode, so to finally take part, it's been a real pleasure.
That's fantastic, and it's a pleasure to have you as well. Do me a favor, take a couple of minutes and tell the audience a little bit about yourself, your work, what you do when you're working, what you spend your time on, and then what you do a little bit when you're not working. One interesting thing people would want to know about you that might surprise them.
Okay, so thanks. So I've been consultant hepatologist in Leeds now for almost 5 years, and I spend half my time in the clinic looking after patients who have complications of end-stage liver disease, particularly liver cancer, and preparing patients for transplantation. We also have a big secondary care practice, so we look after referrals from the whole of the city of Leeds, which is a city of about 800,000 adults, and we deal with all of their liver issues from sort of minor abnormalities in liver blood tests all the way through to advanced disease. And then in the other half of my time, I spend that in the university studying the outcomes of patients who've got liver disease and thinking about the best way that we might improve those outcomes, and also the best way that we can get people into the clinic, so the way that patients can be diagnosed. And that's really been a focus of what I've been doing for the last few months, and something that we'll perhaps talk about a bit later on. Surprising things about me? Well, I don't know. I think maybe it's that I spend most of my time listening to podcasts while on an indoor rowing machine. So it's a bit of multitasking, keeping fit and keeping up to date.
You'd have no way of knowing this, but until the pandemic made me wonder how safe gyms in the U.S. were. Concept 2 rowing machines were my major source of exercise. So that would make two of us that are odd together. Anybody else want to share that particular avocation on the phone? Anyone else here who does rowing machine exercise?
I love to row. That's my warm-up before I get on the bike.
Ian, that's great. Thank you very much. And now we know a little more about ourselves, not just you, but about ourselves than we did before this as well. That's great. All right, moving on. First of all, thanks, audience, for continued interest. You might have received a message through your LinkedIn last night, a special message which was part of a test advertising campaign we ran. If you received it, I'm hoping you clicked back. Thank you for that. Our click rate was 35%, which is crazy high for that kind of thing, and speaks to the idea that there's an interest and a thirst for the kinds of things that we talk about on this podcast. That's exciting. And listenership projections keep growing. I talked last week about our AASLD coverage and what we might get to this week, 2 small changes in that. The follow-up session will wind up taking place on Tuesday, not on Wednesday, which means it will drop the next week at the same time. So for those of you who remember how tired we were by the 3rd straight day of EASL, we're going to do 4 straight days this time, except for Stephen, whose wife's birthday falls in the middle of it, and we'll be taking a night off to maintain the quality of his marriage. And we'll have guests with us every night as well. We will not start announcing who those guests are until next week. And next week, I expect we'll probably be doing our first preview episode. More on that to follow. For now, we're going to keep music on hiatus because we've got too many episodes and other things to focus on. I think we'll try to figure out when we get to the end of the year how to bring music back in January in a meaningful and compelling way. And that's it. All right. So this week, we're going to go back to what we've been doing for a little while, which is you can talk about a personal or professional highlight, whichever one you like best. Please go ahead, brave one first.
Hi, it's Louise here. I'll go first. I suppose my professional highlight is this week we've had our equipment upgraded to smart depth, so we can do all of the continuous cap and go for the extra depths in the larger patients, shall we say, with the rounder shape. So I saw it in Paris last year, so it's now exciting to be able to get my hands on it in the flesh to take it for a spin.
Excellent. That's great. Don, I fouled up. I realized I should have let you make your fashion lineup news of the week, but I didn't, so you have to come up with something else too.
Since I am not a professional fashion model yet, I will have to make do with having been elected to the board of the Council of Medical Specialty Societies. And so very honored to serve both patient and medical leadership in transforming healthcare in that way.
Hear, hear. That's fantastic. Stephen or Ian, whoever wants to go next.
Sure, I'll go. This is Stephen. So I elected myself to the board of Ole Miss football. Can't stand throwing 6 interceptions against Arkansas this past weekend. So that was my low light, personal low light for the week. But the professional highlight would be the fact that I was able to navigate the insane amount of video recordings and presentations that had to be done by Thursday close of business Eastern Standard Time for AASLD. Literally a month before the meeting starts, everything had to be done. Posters that required 5 minutes of video recording, oral presentations that had to be 10 minutes long to a T and had to be re-recorded if somebody opened the door, an email came in or something happened, or you just messed up. And then all the lectures, which normally normally we work on until literally the day before, and sometimes the day of, all had to be done and recorded. So I'm done with it. I can say I think it was successful. We'll see how they're received, but at least I met the deadline. I'm happy about that.
That's excellent. Ian?
Yes, I think the biggest highlight of the last week has been we finally completed the analysis of every single liver blood test that was done in primary care in Leeds in years 2018 to 2019, which is about 350,000 tests, which is a massive undertaking, but fascinating data and really interesting to see how that bears out in terms of what happens to those patients in the long run.
If we get a chance on this podcast, or if not at some later time, love to learn more about exactly what that is and what you're doing and looking for it, because it sounds like an extraordinary effort. I keep teasing everybody. My professional best is only 95% of the way done, up from 92% last week. My personal best would have been for soccer fans, football fans, the return of Gareth Bale, but that turned into 6 different kinds of disaster, as Louise knows, and I guess my personal best is that when Lanzini put the goal in the 94th minute, I slammed my telephone on the couch. I did not break my telephone, thereby showing a very, very small modicum of restraint on what otherwise was not the best hour of my week. Actually, except for that, nothing even slightly untoward happened all weekend. I guess I can say this now. I celebrated the wedding anniversary last week. I'm not going to talk about it any more than that, but that was a delightful weekend and really just a great time. My wife is exceptionally private, would not like me even to have said that, and certainly no more, so we'll leave it there. But with that, why don't we go on to our episode? Basically, what we're going to do is this. We worked out a series of questions for Ian, really about what practice in Leeds is like right now as he described his practice. He'll take a couple of minutes and answer each question, and then the rest of us will follow up with more questions, and then we'll go on to the next one. So just to start, what is the focus of in-clinic treatment of fatty liver patients today? How do they find their way to you? What do you do to establish diagnosis? And then what do you try to achieve in therapy? What are you trying to get done, and how?
So that's a good question, and I think things have changed a huge amount over the course of the last 6 months for obvious reasons. And we have had to make difficult decisions about how we manage patients in the clinics and which patients are seen in person and how patients are followed up remotely otherwise. So looking back 6 months ago, we had a sort of quite inclusive referral policy, so pretty much anybody who had abnormal liver blood tests could get an appointment in the clinic to see us. We had a special dedicated clinic for people with elevated ALT who were at risk of fatty liver of all sorts, and that pathway works quite well. Referrals were increasing steadily and quite quickly, and so we were at the point of thinking about how we might better prioritise those patients coming into the clinic. Those that did get into the clinic were risk stratified using, at that time, NAFLD fibrosis score and then FibroScan, and those who had evidence of fibrosis progression were put into a dedicated fatty liver clinic where patients were managed with the aid of a specialist dietitian to try and aid lifestyle modification and weight loss, and those patients who we thought might be eligible for clinical trials were screened and underwent liver biopsy. And those patients where there was uncertainty because of the limitations of the non-invasive testing also would have liver biopsy if we thought that that would benefit them. In terms of therapy, we're not using any specific medical therapies. I know that other people are using a bit of vitamin E, maybe some of the glitazones, but we've not really done that, so we don't see the evidence really supporting those. So patients have been managed exclusively with diet and lifestyle modification, surveillance for progressive fibrosis, and for those people who've developed cirrhosis, then surveillance for complications. So that's where we were 6 months ago, and I'll be happy to take any questions on that before we talk about how things have changed in the interim.
I was reserving my questions until to see the change. I love the compare and contrast. The top issue always on my mind at this point is helping clinicians weigh the various risk factors and who should they send for screening. And I'm just fascinated to hear the algorithm or steps that you have developed to segment patients and put them each in the right paths, because the Global Liver Institute, we talk about a solution for every stage and wanting to make sure that each patient, no matter where they are along the spectrum of fatty liver disease to NASH, has some form of intervention, whether it is access to behavioral support or dietitians or something more intensive. And so I'm listening very closely to hear the thought process that you've gone through to help place the patients in the proper segment?
I mean, I think that's a really important question. And one of the reasons that we've been looking back at all of the tests that are done in primary care is because we really don't understand what it is about testing in primary care that drives people into the secondary care clinic. We know 35% of the adult population has had a liver blood test in Leeds in the last 2 years, and we've seen a very tiny fraction of those patients, and it's very likely that we're seeing some of the right ones but a lot of the wrong ones and not all of the right ones. And so trying to understand what it is that makes the primary care physician request liver blood tests, what it is that means that they often don't respond to abnormal tests or don't recognize the risks of patients is a big focus, and trying to engage them so that we can, help them do the right test in the right patient at the right time is a massive part of what we're trying to do.
Ian, this is Stephen. So just to follow up on that, I've been mulling over in my mind, how do we keep it simple? The primary care docs, at least in the U.S., have these measures that they're accountable for each and every visit. We call them HEDIS measures, whether it's blood pressure, A1C, colonoscopy screening, mammogram screening, checking the lipids. You have 15 minutes, maybe 20 minutes with a patient, and you're trying to get through all of these things, plus ask them how they're feeling. And it's not uncommon for a primary care patient to, you ask them how they're doing, and they literally unfold a list a mile long that has all of their current issues that they've been sequestering between their last visit and this visit. And literally, you could spend the entire 20 minutes just reading the list. So having said that, how do we make it easy for a primary care physician to identify at-risk NASH patients? And one of the things I've contemplated in my own mind, and I'm interested in your opinion, is when we look at patients with metabolic syndrome, or quite frankly, diabetes, the prevalence of fatty liver is between 70 and 90%. In our study in San Antonio, the prevalence of metabolic syndrome fatty liver is 90%, in diabetics it's 70. And of those, 46% have biopsy-proven NASH. So take it down to 40% if you're conservative, take it up to 50% if you're Ken-Kuzi. But at the end of the day, it's almost flipping a coin as to whether or not they have at-risk fatty liver. And would it be simpler to just say, if you're a diabetic, you have to pass through our screening process, whatever that may be. Maybe it's a set of liver enzymes plus a FibroScan. Maybe it's NAFLD fibrosis score. The problem I have with NAFLD fibrosis score, however, is that when you screen a broad-based population where the prevalence of advanced disease is low, you have a very, very low positive predictive value. And if you're using that test alone, I wonder if we'd be better served by just taking a simple lab test like AST and combining it with something like a FibroScan, just as that initial pre-screen. And then if they're a diabetic that has fat but a KPA less than 6 and a normal AST, you say, hey, go back and see your primary, come back in a year, maybe 3 years for another exam. And then if you're positive, you move over into my fatty liver clinic where we do the rest of the workup and consider maybe a study or more intensive therapy with a lifestyle counselor.
That raises a lot of interesting points and questions. I mean, the average length of a primary care consultation in the UK is, the appointment length is 8 minutes. So if you can think about trying to squeeze in even half of what the patient wants to talk about in that time, it's a big challenge. And from what we've seen and what we know about testing, the assessment of patients for the presence of liver disease of all types is not due to a lack of liver blood testing. If you look back through people's records, I'm sure you see the same thing, that everybody had their LFTs measured at some point. They've always been done. So it's about recognizing who's really at risk and providing, as you say, a better tool to the primary care practitioner to use in the right setting. And whether that is a very simple non-invasive test, AST-ALT ratio or FEV4, or whether it's a sort of imaging-based elastography test, as you say, I think is an open question. There are a lot of patients to think about screening with elastography. So with a very good negative predictive value, you can exclude significant or progressive fibrosis in probably, depending on the age of the patient, maybe 40% of people without the need for elastography. So it probably makes sense to have a stepwise algorithm, but say to the primary care physician, look, we know these are risk factors for liver disease. Forget the liver blood tests. They don't really tell you anything unless they've got a new different acute hepatitis. Just do a fibrosis test, because we know that fibrosis is prognostic, and let's think about how you would stratify the patient into care based on their risk of future liver-related morbidity and mortality.
I've just got a question for Ian and maybe for Stephen, is that if we say that a lot of primary care physicians don't pick up those abnormal tests and put them through the clinics, although I have a feeling if we picked up all of the abnormal tests, our clinics would be swamped. But is there a way to backread the primary care records? There's a lot of computer algorithms and various companies that can go in now and do that to pick out the patients with the highest risks. So to cross-reference it to diabetes, heart disease, high cholesterol, hypertension. So to make that work easier for primary care now to target the right patients to send to your clinics.
So you probably wouldn't even need to backread the record, because the risk factors are so easily identifiable. Diabetes, obesity, we're talking about liver disease, you can't forget alcohol consumption too. So having a way of automating a flag that says this person's at risk of liver disease, have you tested them for liver fibrosis? Yes, no. Then that would be a good way of sort of digging into that at-risk population. The other way of doing it, which requires a bit more sort of proactivity, but probably more of a learning response from the practitioner, is to put that question up front and say, you're requesting liver blood tests. Is this person at risk of liver disease? Yes, no. And that drives them to engage if you've got that in the focus of why the request is being made. But I think both approaches would be worth testing, I think.
I think, though, we need to get it to a sense of what is the relative risk and what's the time frame for that risk. And what I love about the sort of elegance of Stephen's solution and that focus on sort of immediately at risk or urgently at risk, I think we still want to find a way, backing up to sort of 30,000 feet from an advocacy perspective, if you have a liver, you are potentially at risk of some form of liver disease and need to be focused on your liver health. I know that is not yet universally shared. So how do we, and because the risk factors and drivers for different types of liver disease are more numerous than we are willing to talk about in most fora, it really is about how do we weight the different risk factors as they play out in an individual's life. And so what is the time frame of risk to either liver outcomes or cardiovascular outcomes or mortality outcome, otherwise known to normal people as death, that we're talking about here, so that patients understand what sense of urgency and what level of change that we're asking them to take on. Is it just something to be on the lookout and move more, or is it you need to really you need to lose 100 pounds, you need to stop drinking completely, you need to make sure you don't take NSAIDs time, what is the relative risk, what are the weight of all those risk factors, and what's the urgency that we're bringing to the conversation, and I think we don't have that information yet with a sense of clarity that makes it actionable at this point.
Depends what you're asking the patient to do. I'd be interested in Stephen's view, but we do know what the absolute risk of liver-related events is by fibrosis stage. And individuals who've got less than F3, so without advanced fibrosis, in the medium term at least, are at very close to zero risk of liver-related morbidity and mortality in NASH. So you'd have to be a bit careful about using those numbers if we want to, as you say, treatments at all stages, because if you say to people, you're not really at risk of liver disease outcomes now, what provides the driver for change? Is it the associated comorbidities, or are we saying that, we just don't need to focus on liver disease at the minute, come back and have another test in 3 years' time and we can reassess things?
Yeah, maybe we're trying to make it too complicated yet. Still, if you said, look, primary care, and you're diabetics, get a set of liver enzymes, get an ultrasound. If the liver enzymes are abnormal, and the AST is equivalent to the ALT, that patient has a 9-fold increased risk of advanced liver disease. Send those forward. If you get the ultrasound and it shows a shrunken nodular liver, maybe a dilated portal vein, send them forward. Everybody else, tell them to lose weight, exercise, manage their comorbidities. We'll come back around when we've dealt with all these patients, and we'll pick the next low-hanging fruit phenotype. But for now, just take your diabetics, get an ultrasound and a set of LFTs. 2 things at that point: AST-ALT ratio of 1 or shrunken nodular liver. Send those forward. Is that too simplistic, or is that doable, you think? That's not just for Ian, that's for Donna as well in the U.S. What do you think?
I think it's doable, but I think we've missed, and I think I agree with Donna on that, there are associated risks of poor liver health that occur before fibrosis stage, the type 2 diabetes risk, the cardiovascular risk. So if we only ever look at fibrosis score, and I totally agree that if you look at the mortality and the liver-related outcomes, fibrosis is obviously the biggest risk factor for that. But I think it's the other associated diseases. And there was a startling piece, I think, in the Lancet a couple of weeks ago by Shear et al. And the non-communicable diseases, which we talk about when we talk about the associated conditions with NASH and NAFLD, are on course to exceed $47 trillion and cause 30 times more deaths than HIV over the next 20 years. But these diseases receive 17 times less funding than HIV. So in the next 20 years, if we don't get something that we can start to use now, we are not turning non-communicable diseases around. We are not going to improve liver health and well-being if we always target F4 and F3 just for liver disease, but forget the associated conditions that could just be simple steatosis. I don't think there's anything simple about steatosis now when we look at those comorbid conditions. So I think we do have to make it simple, but we have to make it a very big simple.
So let's step back and posit a couple of things, because I think it'll help. Number one of the things Ian was saying in our pre-conversation that I'm just going to bring up because I don't think we need time to dig all the way through to, is that virtually everything that you'd focus on if you're in a hepatology clinic is the late-stage fibrosis patients where you can have immediate impact on the liver. That said, there's a silo in medicine, which is the people treating non-communicable disease tend not to be hepatologists. Right? We've had other folks come on, other European key opinion leaders come on and flat out say, well, listen, I'm here to treat the liver. In the States, maybe philosophically we're a little better, but practically not. So, Louise, one of the challenges to what you're describing, and I think it's critical, but I'm not sure how to do it, is how do we start to break down the silos between different specialties and different views so that we can see where the big issue is in the smaller questions? And then, by the way, how does that affect, and this is to Stephen or to Ian, how does that affect what a hepatologist would do for a living after we do a better job of breaking down the issues around communicable disease, as compared to what do we push back upstream to primary care and other specialties?
I think breaking down the silos is going to be difficult. I think Donna and GLI, and it seems to be a bit more sort of coordinated within the US with endocrinology leading a little bit more. But I think we've got new medications coming. We've discussed some glutamide, which is a diabetic medication. There's been an awful lot of discussion within the diabetes forum. We know that it helps within NAFLD and NASH, so maybe we've got a drug that at least 2 associations that combine for guidelines actually are quite excited about, which might make us sort of coordinate, because I doubt we're going to have 2 different ways of treating and using those medications through 2 different types of pathways that become complicated or not succinct if it gets FDA approval in the coming years and gets through its trial status. So maybe that's the excitement between a little bit more coordination, but I don't know. At the moment, silos are very hard to break down.
They are hard to break down. And one of the things that I say to my trainees when they come to the clinic is treat the patient, not the NASH. And I think that this idea that there'll be therapies that will have multiple effects on diabetes, weight loss, cardiovascular risk, like the GLP-1 receptor antagonists, which will probably also improve the NASH, it's likely for a lot of the patients who are treated with those, it's those other endpoints that are going to be most important. And whether really hepatologists are the best people to be treating large swathes of that population of people who've got diabetes is really an open question. And I think we probably need to be more inclusive and probably a bit less focused on the NASH and say, actually there are people around who've got medicines who are going to help our patients. Let's work together towards a solution rather than really just honing down on the NASH.
Okay, so And by the way, that's before we bring this whole issue of COVID into the picture, right? On two levels. Number one is, as Ian said, things are different than they were 6 months ago. Second, as we discussed on the podcast a couple of weeks ago, we're starting to see data that suggests that the relationship between liver and COVID might not be, say, like heart, where the organ is damaged by the disease and independent of it, but where actually liver fat and obesity become predictive risk factor. So you get those two issues, which is now you're treating differently, and maybe we have to think differently about how we're treating in a way that's even more inclusive of all those silos we're having a hard time breaking down. So Ian, what's different now than the picture that you presented about what life was like for you, and the picture you presented about what life was like before COVID? Let's start there.
Short answer, everything. Long answer, we've made major and quite radical changes to the way that patients are referred into the service. So having gone from a sort of widely inclusive referral pathway, we've moved towards a pathway that is very focused on identifying people who are at increased risk of liver-related morbidity and mortality. So done that through implementation of a referral pathway that mandates FIB4 testing, and if that is not low and reassuring, then the patient has an ELF in primary care. And we've done that to parallel the way that the cardiologists look after their patients, and so they look to see what the patient's risk is, then they think about providing primary prevention of cardiovascular disease and cardiovascular events. That's had a big impact on the number of patients that are coming through to the clinic, and then when they come to us, because of limited clinic space, because we're allowed few patients to be seen face-to-face for social distancing reasons, patients are going to a satellite hospital, having elastography, repeat blood tests and an ultrasound before they're coming into the clinic. So we have now a sort of much more streamlined and selective pathway for patients getting into the service. And I think that that's likely to persist. It's become a necessity because of COVID, but I think the efficiencies that it provides to secondary care are sort of obvious to my colleagues. There still has to be a focus on primary care, as we've talked about getting the right patient to the right test at the right time, so that that streamlined and efficient service that we're seeing isn't providing a barrier to patients being referred in those patients who need it being referred in the first place.
So I'm just kind of looking at my colleagues, really. What does that tell you about what's likely to get better or not as the real healthcare system tries to cope with this issue on top of all those other issues?
One thing that's really exciting, I think, about the type of transformation that we've had to undergo in healthcare due to COVID is that every clinical practice has had to go through their patient panels, patient by patient, and be able to assign them, is this urgent? Can this care be delayed? Can this be done by telehealth? Do they need to come into the office? And that type of assessment really has never been done before. For all we talk about population health algorithms, have maintained for years that we can't get to a healthy population without healthy individuals. And the work of looking through what do individuals need for care had never been done before COVID. And so I think that we're never going to go back, or I hope we do not go back to a state where it's just sort of a conveyor belt of patients anymore, but that some of that personalization and that segmentation in where care is delivered, who delivers the care, and how and what care continues to move forward. I think that's particularly applicable to patients with multiple chronic diseases, liver disease certainly, which is complex in and of itself, and certainly applies to NASH. So I think care has already been disrupted in a way that I think will have positive effects for years to come.
I use correct to say, did I hear that? So, there is brilliant data by your colleague at Newcastle and others, even Phil Newsom's done some work down in Birmingham looking at these sequential tests, and others, many others have looked at this as well. And I actually think there's data, I can't find it in my brain right off the top of my head, of FIB4 plus ELF. And it's something like, I think the AROC for that is like in the 0.7, maybe 0.8 range. It's pretty good, actually. What I would be, and I assume you're using 9.8 as the cutoff for risk?
Yeah, we're sort of exploring cutoffs. We've started with a slightly lower threshold to be sure that we're not missing significant disease. We're at 9.5 at the moment, but as the cases are coming through, I think it's likely that we will go up a bit, probably to 9.8, if not even a little bit further.
Are you chasing them with liver biopsies if their FIB4 is indeterminate or positive and their ELF is higher than that number?
So they come for a FibroScan after they've had their FIB4 and their ELF. ELF as the sort of arbiter test. And then if there's continuing uncertainty, then where we think it's going to provide useful clinical information, then we would do a biopsy after that.
So, you just gave me a bunch of additional information I didn't either hear or know about. So now you're actually adding a third non-invasive test. You do 2 sequential blood tests followed by an imaging study. Am I hearing that right?
That's right, and we're doing the ELF to try and keep people outside of the hospital environment. So within primary care, not needing to travel into the hospital.
What percentage, do you know a number off the top of your head, what percentage of indeterminate or positive FIB4, positive ELF actually they go on to FibroScan? So let's say 100% of those people that have indeterminate FIB4s and positive ELFs get a FibroScan. What percentage of those have a KPA above 8.5?
So I can't tell you that off the top of my head, and it's still a relatively new innovation because it's only been introduced over the course of the last 6 months with COVID really impacting on the service. So it's still quite new. The data are coming, but I don't have them on the top of my head yet.
So 2 quick things. I would love for you to submit an abstract to NASH-TAG. And if that's too early, please submit one to EASL for Amsterdam, where you're looking at several things. Number one, the demographics of the positive population. What percentage are diabetics or obese, have metabolic syndrome? And then number two, how is that translating into risk stratification for you? In other words, play it out all the way to the end. Are you really super selecting for that positive patient that you can meaningfully impact a change?
That's what we need to understand from what we're doing, and particularly whether the sequential ELF and FibroScan approach is adding a lot of information, because it is adding significant additional costs. So we are working on all of those questions, and I hope that that'll be coming to a meeting near you soon.
Maybe I heard this wrong. Okay, again, going back to my last science course was in high school biology, but Stephen, if you say that the AROC for FIB4 plus ELF is the one that gets to an attractive enough 0.7, 0.8, an attractive enough level, and Ian, you're looking at, can we do away with one of those tests, or do we do them which is kind of what I think I just heard you say, can we get away with only one of them?
Yeah, I think, just to clarify, Ian, you mentioned you guys are doing FIB4 followed by ELF. Is that sequential non-invasive testing strategy correct?
That's what we're doing. Whether it's correct or not is another question, but yes, that's what we're doing.
Okay, so Stephen, do me a favor, off the top of your head, square that circle. Would you expect all that to fall out? Just guessing.
Well, certainly we know sequential testing is better than individual testing. The real question becomes, and we also, let me say, I think in my own mind that a wet biomarker plus an imaging biomarker probably is going to be our best option. So really it comes down to what's better, ELF or FIB4 as the wet biomarker of choice. FIB4 to me is a free test. Nobody has to, you get the liver enzymes and the platelet count, but age comes free. Well, that comes at a cost too, but that's a different
I was about to say, yeah.
different story.
I'm older than y'all. Yes, it does. Go ahead.
I noticed that when I crossed the 50 barrier not too long ago. I think certainly I believe FIB4 is probably less expensive, at least in the U.S., I suspect in the UK and other parts of the world, than ELF, which in our part of the world runs about $400 and takes about 2 weeks to turn around. You can order ELF in the U.S., but it's a specific send-out lab to Siemens Healthineers in California. So it's not like you can order it through Quest or LabCorp. You literally have to go to the Siemens Health website, download their address and how to, draw the blood, what tube to put it in, and ship it. And then you wait for the answer to come back. So if we could find a way to make FIB4 that preferred test, that would be easier for primary care to wrap their hands around, at least in the U.S., at least today. The problem will always be that it has a better negative predictive value, and I suspect that holds true for ELF as well. All these tests have better negative predictive values than their PPVs. That's why we link them together to improve the PPV. Unless you're testing a high-risk risk population. Let's say you're in the portal hypertensive clinic, and then, you have your FIB4 will have a very high positive predictive value. So I don't know we haven't answered your question, Roger, but it is, what Ian is doing is unprecedented, and it is highly valuable, not just for his community, but for every other community. And if you're listening to the podcast, you want to stay tuned to what Ian is doing, because he's on the cutting edge of this thing.
I have to say just now that it's not just me. I have a colleague in Leeds, Richard Parker, he's been driving a lot of this work forward together with me. He deserves due credit for this work that's being done.
Fantastic. Let me shift gears. There are one or two other questions I want to get. So, Ian, you've listened to all 30 episodes, and you're ground level treating patients. What that's come out of this podcast, if anything, do you see either in your world or in what you see around you as having an effect? Which of the issues that we've discussed on this podcast do you see as having any effect on changing how anybody's thinking about treatment right now? And the answer might be none, by the way.
I think to talk about that, I think you probably have to understand at least my perspective, and that is about how treatment might be used in practice, because ultimately that's what really matters to me and the patients that we see in the clinic, and what is a new therapy going to mean to them. And I think that a lot of the discussions that were had around the complete response letter to Intercept about how a drug might come to receive a license and what it would need to show in trials to do that, and then the later discussion that was, I think, perhaps in the digital ILC wrap-up about what is it that a treatment's going to achieve. Is it around treating only liver disease? Is it a treatment that's likely to have broader metabolic effects with also an impact on liver disease? And those sorts of discussions are becoming more and more relevant as we try and understand which patients we should think about selecting for treatment and how they'll be treated. And we've sort of touched on liver biopsy, but we've spent a long time talking about how patients are being identified in the clinic and how and whether those two strategies are compatible. If you've got a treatment that's been developed based on a liver biopsy selection which shows NASH of a certain severity with a defined fibrosis stage, when in reality in the clinic lots of the patients are really just going to have fibrosis testing, and we'll never really know how bad their NASH is, and there's not really an indication to do a biopsy without access to clinical trials for a lot of those patients. And it's that sort of discussion that I've personally found most interesting, and it's begun to change the way that I think about how treatment might be delivered in the clinic.
Do you want to elaborate about 3 sentences more on how that is? You see it might develop?
So I think that development of tests that might replace liver biopsy, that's the area where I'm beginning to think that we might be going. Understanding how we'll use those based on the information that will come from trials where patients were selected on biopsy that's the foreseeable translation gap for me for seeing patients in the clinic this year, next year, about how we'll get access to treatment for those patients and how we'll know what benefits it'll give them when those patients may not be exactly the same as those patients who went into the trial.
That's really interesting. Stephen?
Ian, this is what we're working feverishly on right now, and probably the biggest, there's a benefit to developing drugs in NASH that have to be linked to a liver biopsy. Donna just cringed when I said that.
I did.
I know you well, but you're gonna appreciate this comment. So paradoxically, this is giving us the ability to move non-invasive testing ahead at light speed, because we're collecting the data on most every phase 2B and 3 trial out there. We're collecting non-invasive testing data, both wet biomarkers that don't exist yet, for which we are storing serum, and imaging, not just FibroScan, but I'm talking PDFF, I'm talking MR elastography. And then on top of that, you've got the Litmus and Nimble consortium that are looking at this through a very pragmatic way. They're taking preclinical models, studying them. They're taking what's out there and assessing them. They're comparing it to baseline liver biopsy. And when the imaging modalities come into play, we're not just looking at MRI, PDFF, and MR elastography. We're looking at DeMilli. We're looking at diffusion-weighted MRI, lots of other techniques. But I'll tell you, the sobering fact we're at today is we've learned more about how they're not useful and about what the limitations of the test are. are, then how we should actually be using them and how we can improve the precision and the accuracy of the test. And of course, we are hindered to some degree, maybe would someone argue to a big degree, by the variability of liver biopsy and by the histopathologic inter-observer variability of the read. And so I think we're at a crossroads where we have science and technology to give us ways to measure fibrosis turnover of the net collagen activity. We have ways to measure, to a lesser extent, inflammatory activity, and to even a lesser extent, probably ballooning. But collectively, we have ways to get after this. But we need everybody to come together and really look at this and, determine what the best way forward is going to be. Because I think in 2020, we've learned what our limitations are to these tests, and we've learned that putting them together in sequence gives us a better precision and accuracy, but we haven't figured out the exact right way to do that. And I think that's what we're going to unlock with a lot of these clinical trials. And so, we need the liver biopsies right now because I do think it allows us to study non-invasive tests in a way that we wouldn't be able to otherwise. But this is also where artificial intelligence and machine learning will come into play to help us bridge that gap of uncertainty on liver biopsy. And that will solidify these non-invasive testing strategies moving forward. And I predict will put us into the 0.9 range on precision and accuracy. So it's not just a liver biopsy read by expert hepatopathologists, but it's liver biopsies read by an expert hepatopathologist and adjudicated with machine learning, and then non-invasive tests are measured against that.
Stephen, I appreciate your thoughtfulness on this, and I do appreciate how close we have gotten in terms of during the course of recording these podcasts and advocating in the field of liver health, that my reactions without having to look at them. I do want to state that I understand the value of biopsy. I think the role of advocacy is to encourage researchers and clinicians and regulatory bodies to challenge the assumption that biopsy is needed and to explore those limitations, to admit those limitations, to put a time limit, nay, an expiration date on biopsy rather than just to perpetuate it because that's the way it has always been done in the past. And so, what I am so excited about in terms of, and what I value about the NIMBLE Consortia and the Litmus Consortia and all of those who are working particularly in a collaborative fashion in this field is that we recognize that there is a process that we need to accelerate, that there is a point where we need to move past, I won't use my own hashtag in this, but we'll move beyond the biopsy, we need to move past this point at some period in time. When I first started, it was talking about decades, and it'll always be a use for it, and now we're recognizing that there's a limit to the usefulness of biopsy, and it's about how and when do we get past that, and that's the conversation that advocacy causes people to have, and by which I measure my effectiveness. And so I'm excited to see and gratified to see the progression of the conversation about the contexts in which biopsy is useful, if any.
Okay, Stephen has a hard stop in 3 minutes, so I'm going to ask him to answer the last question, and then I want to ask Ian a question. I want to make a comment on what you just said, and Louise might have done it also. Stephen, one thing that surprised you most that you heard this week?
Well, I am just amazed that Ian and his colleagues are doing what they're doing. I think that sort of real-world data is sorely needed for all of us. I just am thrilled that that's happening. That was a huge surprise that you're actually doing FIB4 followed by ELF and subsequent transient elastography where positivity is found on the two wet biomarkers. That's phenomenal work. I can't wait to see how that plays out relative to the endpoints that you're hoping to measure. So you made my day there.
Donna, response to your comment, and then Louise, if you have anything, and then I have a final question. I want Ian to get the last word today, and then we'll go back to the roundup. It seems to me when I listen to what Stephen's talking about and what you're talking about, even what Ian's talking about with the work that he's doing, that the question around biopsy is going to be less of a deadline and more of a finish line. That the ability to improve biopsy through AI means we can learn a lot more from it at the same time that we're doing a much better job on wet markers and diagnostics and mechanics of non-invasive testing, means that we can get to a point where we know enough that we can confidently live without it. And to me, the great thing that you've done is created urgency around getting to the finish line as fast as possible. We're no longer talking about walking or even speed walking or even marathon. We're now talking about how fast can you run a 5K, because you've got to run at that kind of pace, if not faster, to satisfy all the challenges and issues that you've identified when you talk about beyond the biopsy. Does that make sense to you?
Yes, absolutely.
Yeah, okay. Okay, good. Louise, you have anything you want to add before we give Ian the final word for the day?
The only thing I had to add was I knew when Ian and Stephen were talking that I'd seen something at the Digital ILC on serial tests, and it was a non-invasive test for assessing fibrosis in patients with NAFLD, an evaluation of combining test results, and it was Catherine Vick and her team. It's poster number FRI009, and it's a very good poster because it does look at FIB4 followed by ELF. Done in 3 studies. So what they did was calculate the specificity and the accuracy of those tests. So that might be a good poster to look at for some of the bases if you've not seen it. It was quite an interesting one and did lots and lots of various combination of various wet markers followed by FibroScan or things. So that I knew I'd seen something about some of the data that Ian was looking to produce when I was doing Digital ILC, and I think it's a fascinating project and it'll be really interesting to see that because, as you know, I think FibroScan's in the wrong area currently, which makes it very expensive. But I can't wait to see what your data produces, and if you can get it out by NASH, great.
So, Ian, last word right now looks like this. You've listened to 30 episodes. You do what you do. You were talking about today, and frankly, I didn't know when I invited you on that you were going to talk about this piece of research you were doing that was going to light Stephen up like that. He lights up like that about maybe once a month, maybe not that often on this podcast. What is the thing we should take away with us from your experience that gives us the greatest optimism and then conversely the greatest concern about the challenges that hepatology is facing in treating patients in this environment right now?
So I think the nature of the pressure that COVID has placed on health services has led to much quicker implementation of referral pathways, telemedicine, and other changes that I don't think that we would have seen without that stress. And I think particularly for patients who are at risk of having significant liver disease, that additional testing in primary care is likely to identify those individuals more quickly and in a way that is perhaps more acceptable to the patient in the first instance. So I think that stress that's been provided in the system has really led to a degree of innovation that we wouldn't have seen otherwise. The associated challenge is that there are still barriers to implementing the treatment and management that's needed for patients who are experiencing delays getting into the hospitals for whatever reason, and we're still catching up from the first phase, and we're the second wave is on us, and we'll be catching up again after that.
Okay, and that's a great note to close on. Thank you very much for that. So, last round, Stephen already answered. Ian, you know the question because you've heard it often enough. Brave one, go first. The thing that you heard today that surprised you the most or made you think differently than you did coming in?
It's Louise, and I'll jump in on that one. I think the thing that wasn't surprised me the most but impressed me the most is the coordination and the results from primary care, because I think that's, as Ian said, is where we're heading back to trying to get better quality and better diagnostics into primary care. I worked with the shared care system within Australia because of the remoteness, and I think they do work very well if you have the ability to perform a lot of the fundamental investigations where the patient wants it most, which is usually where they live. So I think the work that he's doing gives me a great deal of joy and hope.
My favorite takeaway was treat the patient, not the NASH. I hope everybody takes that to heart.
That will be our line for the week. That's right. It seems we have one of these every week, and that will be the line this week. Okay, thanks. Ian, go ahead.
Yeah, and I think the thing that will have the greatest impact on me is that statement from Stephen about the use of all of the information that's generated within the trials to inform the development and assessment of the non-invasive test, and really that activity is accelerating the path towards treatment of patients without the need of biopsy.
And I had an aha actually. It's kind of a major slap your forehead aha as I was listening to this particular episode, which is, as you know, my whole career was pharmaceutical marketing research. And when you spend your time in pharmaceutical, you look at the world through a very pharmaceutical-centric point of view. And one of the things I've enjoyed and been struck by about this podcast is that no matter where we start, we wind up coming back to diagnostics. As if we just don't understand enough yet about how to look at the patient most productively. But there were a bunch of themes coming together in this conversation. One, the one that Louise raised about we need to pull primary care and specialty together to get coordinated care that makes sense, given what the hepatologist's role is and how much non-communicable disease there is back there that we have to deal with. And some about listening to Ian and Stephen's reaction, and then Donna, your comment about what to do with biopsy. And the realization that the best path forward for this disease isn't merely going to be to get better drugs, although that will help, but to get a sounder understanding of what the heck it is we're dealing with and what we're treating and how the pieces go together, so that we can, going all the way to your question, Ian, without thinking about biopsy this way, so that when we get medications, we can be thoughtful and productive about how to best use them to help the most people the fastest with the fewest false starts. I think we didn't know that this year. And I'm starting to believe that the thinking that we're hearing over and over again, no matter where we start this conversation, is we may very well be there in a year or two when the medications start to come along that can bring even more value than what we can get if say, OCA does get approved. So I didn't expect to come out of this conversation this optimistic, but I am. I think that's really a neat thing, and that is where I will close. So with that, first of all, Ian, thank you. That was a better and a more enlightening episode than I even would have bet on when we invited you in, and I've been kind of itching to have you here for a while, and this has been great, and thank you very much for that. Stephen, Louise, Donna, always thankful. Thank you. I feel every week we break down barriers every time we talk, which is really exciting. And then to everybody who helps make this work, Mike Wilson, our engineer, Buzzsprout, Eric, Polly, Ryan, everybody who listens. We're keeping this discussion group up on LinkedIn and Facebook, and people don't contribute very often, but I would love to hear if we've got people listening who actually are treating patients. How does this reflect your experience? What's it similar? How's it different? That's a conversation I would like to get going on the podcast. I'd like to go in the discussion group. I'd like to keep that going in real time as much as possible. We will be back next week. I expect we'll be back talking for the first time about AASLD, a conference for which, Ian, I'm sure your paper will not be ready whenever it is ready. I'm sure in 3 weeks it's not going to make it. And with that, wishing everybody a great weekend and a great week, and stay safe, stay healthy. Surf on. We'll see you back on the podcast. Bye-bye now.
Drug developers, investors, researchers, and corporate executives wrestle weekly to understand what is happening in commercial development of NASH medications. Join hepatology researcher and key opinion leader Stephen Harrison, patient advocate Donna Cryer, liver wellness advocate Louise Campbell, and forecasting and pricing guru Roger Green as they discuss the issues affecting the evolving NASH market from their own unique perspectives on the Surfing the NASH Tsunami podcast.